So students, now today I am going to start the lecture before you, in front of you. So that lecture is the digital lecture class, and you know, all of you have know about that this is the tough condition, and due to the tough condition you have to read at home. So be at home, be safe, and save lives. So now today we are going to start the second chapter, not first chapter. First chapter is cell, and you all of you know about the cell in class. You have read about cell in class 9. Today we are going to discuss the chapter that is structure of chromosome, number 2 chapter. So chromosome, you know, this is the, if I draw on chromosome, this is the coil structure. The coil structure. This is if it's a chromosome, this is called chromatid, and this is called centromere. This is called centromere. So centromere is joined the chromatids in the like a butterfly-like structure. This is called, if I say that what is chromatid, what is centromere, how it is formed. So before that we have to know about some structure of this chromatids, how this is formed. Now we know in our blood there are different types of proteins are present. Different types of proteins. Pure globular proteins are there, and one type of globular protein is present in the blood, that is called histone. That is called histone protein. And this histone protein is a globular structure. And different kinds of histone proteins are there. Different kinds of histone proteins are there. And if the histone proteins are joined together, and 4 are here and 4 are in the back, altogether 8 histone proteins are joined together and make a globular-like structure. It's a ball-like structure. And the DNA, that is deoxyribonucleic acid, that is close over here, and making a complex structure, making a coil structure, super coil structure, and this is formed that structure. That means DNA along with that 8 histone protein, this form is known as nucleosome. So what is nucleosome? A nucleosome is the structure where the DNA along with 8 histone protein, this structure is known as nucleosome. And these nucleosomes are combined with each other and making a chain-like structure, and like this they are known as chromatin fiber. Where you have know about the chromatin fiber? Now in the nucleus chromatin fiber is present, and this chromatin fiber are formed like this and making a butterfly-like structure. And this is from the chromosome. And these are the chromatid, and this is in the middle, that is the centromere, that is joined with the centromere. So here you see the nucleosome, the chromatin fiber, nucleosome ki. What is nucleosome? The DNA along with 8 histone proteins. Now this chromatin fiber are combined with each other, making a chain-like structure. This chromatin fiber is making a chain-like structure, and chromatin fiber is making a butterfly-like structure, that is known as chromosome. Now here I have told about the DNA. What is DNA? DNA is the double helix structure and deoxyribonucleic acid. Now DNA, how the structure of DNA? The structure is like this. DNA structure. Now, if I say DNA structure, what are there in the DNA structure? DNA structure mainly is formed from a nucleotide. What is nucleotide? We have to know about the two terms. Nucleotide. Nucleotide means nitrogen base plus sugar plus phosphate. This is called nucleotide. So nitrogen base, sugar plus phosphate. Another term is there you have to memorize, that is nucleoside. Nucleoside, only nitrogen base and sugar is present. nitrogen base and sugar is present. So nucleotide and nucleoside. So nitrogen base. What are the nitrogen base? Two types of nitrogen base are present. One is purine, another one is pyrimidine. Purine and pyrimidine. In purine, two types of purines are there. One adenine, another one is guanine. Adenine, guanine. Short form is A, guanine short form is G. And pyrimidine, thymine, cytosine, and uracil. Uracil only for RNA. Only for RNA. And T means thymine only for DNA. And C. So, what are the nitrogen base? Number one, adenine, guanine. That is in the purine group, pyrimidine group, thymine, cytosine, and uracil. Thymine in case of DNA, uracil in case of RNA. This is the nitrogen base. Now we are going to know about the structure of the DNA. How it is formed? You see, adenine always combine with thymine. Guanine always combine with cytosine. So like that you can put anything, and these are combined with each other by the hydrogen bond. By the hydrogen bond, and they are joined with 1,1 sugar molecules. That is pentose sugar. This is also known as pentose sugar. Pentose sugar, and that pentose sugar is combined each other by phosphate. By the phosphate. So you say you can think that about A always combined with T, G, C, C, G, A, T. More are there. I am writing here only 4. Now in that 4, if I ask how many nucleotides are present in that 4, you see here one nucleotide, nitrogen base, sugar, phosphate. Nitrogen base, sugar, phosphate. How many nucleotides is present in that line? 2 nucleotides are present. So 2, 2, 2, 2. 2 4s are 8. So altogether 8 nucleotides are present in the small part of this structure of DNA. Now if I draw like this DNA, sorry, this is not like this. Okay, now if I draw like this DNA, the nucleotides are present like this way. The nucleotides are present like this way. And if I start from here, the circle ends here. One circle ends here, and this is altogether the length is above the 35 angstrom. 35 angstrom, and here altogether 10 base pair is present. 10 base pair. 10 base pair means 1, 2, 3 like this. 10 base pairs. One base pair means like this. One base pair. 1, 2, 3, 4. 10 base pairs are there. So 10 base pair is equal to 35 angstrom. So 1 base pair is equal to 35 by 10. That is 3.5 angstrom. So 1 base pair, the distance between base pair is 3.5 angstrom. And this is the original structure of DNA. That is a double helix structure. Supercoil structure is called also supercoil structure. Next day we will discuss the next part of the DNA and the cells. Okay. তুমি শুনলে না এটা তোমার কাজে লাগতো?